So my protein was citrate synthase, and it is the main enzyme in the citric acid cycle. It basically starts the citric acid cycle. So I took a couple images from my paper, and I'm just going to kind of explain what went on, or what's going on in these images. Like I said, my protein is the citrate synthase. And here is the citric acid cycle, and here you can see right here is the citrate synthase. So basically, it's going to be taking the acetyl-CoA here and this oxaloacetate, and it's going to be making it into the citrate. And that is actually going to kind of kickstart this citric acid cycle, or the Krebs cycle, or Krebs cycle. So this here is actually the primary structure of the citrate synthase, and this blue right here is going to be the backbone of the enzyme. Here is the secondary structure. These ribbons are going to be the alpha helix, and this blue part here is going to be the beta pleated sheets. So this would be the ribbon structure over here. Here would be the tertiary structure, and all these little green parts right here, those are going to be little protein folds, and then over here, this white part here, oh, right there, that is going to be the actual enzyme itself. And then here in the essay, we were asked to about the gene expression, and asked to give a part of the data marker. So this is actually part of the data marker with the gene being expressed. And there are a couple of diseases that are related to this gene. One of them is critical illness polyneuropathy, and the other is cat scratch disease. So those are just a couple diseases related to this protein. And also some studies were done with this, and whenever this protein the expression of it was decreased, enzyme activity was also decreased. So a few other facts about the protein is that it's an induced fit enzyme, which just means that after it's got two subunits, and so after one of them has attached and it closed, then the other one just attaches to that closed site instead of it attaching like on its open site and then fitting perfectly together like a lock and key. It's a key mitochondrial enzyme. It's found in eukaryotes and prokaryotes, basically found in most living cells. It's, like I said, the first part of the citric acid cycle. It is a first-rate limiting enzyme. It plays a key role in maintaining energy production in different types of cells. It's just a really important protein and enzyme for many different things. And it's used mostly just in the citric acid cycle, and to produce that, to kickstart that, get that ATP produced in order for it to be used in other cycles. So that's pretty much the general summary of my protein, which is citrate synthase again. So